So today we'll be talking through what's basically the start of chapter 13 in your textbook about the nervous system and about nervous tissue. So the nervous system consists of all the nervous tissue in the body. That's the brain, the spinal cord, and the nerves that travel through the body. The nervous system is another system of integration and control. We already talked about one system of integration and control. That's the endocrine system. And we'll be spending the next few lectures thinking about the nervous system. The nervous system is responsible for these swift, brief responses to stimuli by changing the activity of other cells, tissues, and organs. You know from reading about the endocrine system that the endocrine system is also responsible for integration and control, but it's slower to respond and has longer lasting, more widespread effects. So the nervous system, it's all of that nervous tissue in the body, and some general characteristics and function are that it's associated with integration and control of other cells, organs, and tissues. And it's more typically associated with rapid or short-term activities. It's responsible for detecting and processing sensory information. So this is things that you're aware of, like light and sound, but also things that you're not aware of processing, like blood carbon dioxide levels. With all of this sensory information coming in, you're aware of multiple stimuli, and you have to process and prioritize. Then this is called decision making, like when you're driving a car and you have to deal with lots of stimuli at once. So the nervous system is involved with conscious and unconscious decision making, with the formation of memories, and with emotional processing. The nervous system also is needed to excite and control skeletal muscle that's at that neuromuscular junction, and to control all glands. It also regulates and controls smooth and cardiac muscle. The nervous system is divided up into two different divisions anatomically. The central nervous system is the brain and the spinal cord, and this is a processing and coordination center. And the peripheral nervous system is everything that's outside of the central nervous system. So it's providing sensory information to the central nervous system and then carrying the motor commands away from the central nervous system. And here are the basic functional components of the nervous system. So in the outside world, there's some change in the environment. That's a stimulus. And that could be the external or the internal environment, but it's some change. It's detected by a sensor or by a receptor, and that information is relayed to the central nervous system by a sensory neuron. So it's relayed to these integrative centers in the central nervous system. This whole top row is a sensory communication line. It's considered afferent, A-F-F-E-R-E-N-T, is it's bringing information to the central nervous system for processing. Then over in the green are two components of the central nervous system. This is where integration is occurring. This is interpretation of sensory information and coordination of the appropriate response. From there, a motor neuron carries information away from the central nervous system to an effector, a muscle or a gland, and that produces an action or an effect, often opposing that original stimulus, bringing the body back into stable conditions. So this whole bottom row is the motor communication line, or the efferent communication line. That's E-F-F-E-R-E-N-T. So this is the basic functional component of the nervous system. In neural tissue, we find two general cell types. The first one is the neuron. The neuron is the functional cell of the nervous system. It's sending and receiving information. So they're responsible for the transfer and the processing of information in the nervous system. It consists of a cell body, which is called a soma, and that's where all of the organelles, the nucleus are, and then some long projections that stick out in various directions. And these are the axon and the dendrites. And I'll show you a picture of those in just a second. So it's lost some of the characteristics of a typical cell. It doesn't reproduce, and it has to be supported by these supporting cells, neuroglia, or sometimes called neuroglial cells. Neuroglial cells are these supporting cells that protect and nourish the neuron. They can bring in nutrients for neurons and provide protection, help guide repair. And they might also assist with the sending of information through neurons. Neuroglia vastly outnumber neurons within the nervous system. If somebody has a brain tumor, it likely originated in the neuroglial cells because of this high number of neuroglial cells and because mature neurons aren't dividing. So neuroglia, much more abundant than neurons, and they're capable of dividing, which neurons aren't. So we're going to talk through some of the characteristics of neurons first. This is the general structure of a neuron. You can see at the center here, where's my laser pointer? At the center here, this is the cell body. And the cell body looks pretty much like a normal cell. It contains a nucleus with the nucleolus, where the ribosomes are assembled. There are Nissl bodies, these dark gray staining areas that are clusters of rough endoplasmic reticulum and free ribosomes, so where proteins are being assembled. And there are mitochondria, other organelles, and other inclusions, basically meaning stuff, products that's inside of the cell body. Over here on the left side, these small branching structures, these are all dendrites. Dendrites are the receivers, like the antennas on a little TV. So these are responding to a stimulus, and that stimulus could be a touch, a smell, it could be neurotransmitters released by another neuron. So these dendrites are the receptors that are stimulated by environmental changes or the activities of other cells. Then coming out the other side of the neuron is this one long extension. That's the axon, so one single long branch. It's longer than the dendrites. And this is the transmission line for nerve impulses. So we have a nerve impulse, basically the same as an excitation event in a muscle fiber, and it travels down the axon toward the synapse, where a neuron communicates with another cell. So the axon is this transmission line. The axon hillock is this tapered region where the axon meets the cell body. So this axon hillock, this tapered region where the axon begins. The axon can then extend out from the cell body, and the axon can be just millimeters long, or it can be several feet long. You have neurons with their cell bodies at the end of your spinal cord, and then axons that reach all the way down into your toes. That's one single elongated cell. At the end of the axon, they branch into these branching structures that are called telodendria, not to be confused with the dendrites at the other end. And then they end at these enlarged buttons that are called axon terminals. The axon terminals are where the neuron is going to affect another neuron or an effector organ. And down here at these axon terminals, they're right here at this synapse, and they have vesicles filled with neurotransmitters that they're going to release into that synapse. So what we're seeing here is a presynaptic cell, illustrated in blue, and it's sending a message, one-way transmission down the axon through the telodendria, and then the axon terminals will be releasing neurotransmitter into the synapse. That makes this cell right here in purple a postsynaptic cell. Neurons can communicate with several different types of cells. So they can communicate with other neurons. That's the first one shown up here. So the neuron is shown in blue with its dendrites where it's receiving information, the soma, the cell body right here, the axon hillock, and then the axon with this one-way electrical event traveling down the axon and being received by the dendrites of this postsynaptic neuron over here. A neuron can also communicate with a muscle fiber. This is a neuromuscular synapse, or we called it the neuromuscular junction. And then lastly, a neuron can communicate with a gland. So it can have an axon that extends outward, branches into telodendria, and then have axon terminals where that neurotransmitter is released and stimulates these postsynaptic cells over here. So a neuron can stimulate other neurons, skeletal muscle fibers at the neuromuscular junction, or gland cells. A single neuron can control multiple muscle fibers, as you know, or multiple gland cells, or communicate with multiple other neurons, but it was not going to be communicating with a combination of these types of cells. And here's another illustration of this chemical synapse where the neurotransmitter is being released. So up at the top, a slightly larger view in purple is the presynaptic cell with the dendrites here receiving information, the cell body with all of the organelles. Here's the axon hillock, and then a long axon extending down, branching into telodendria, and axon terminals stimulating postsynaptic neurons. So if we looked a little bit more closely, as we do down at the bottom, this is a close-up of one of those axon terminals and the postsynaptic cell. So a message is being sent down the axon, and it's traveling down the telodendria into the axon terminal. And this axon terminal, this is the presynaptic cell. This is going to have the same basic structure as what we looked at in the neuromuscular junction. So this is the end of this presynaptic cell, and this axon terminal is filled with synaptic vesicles. It has mitochondria. It's prepared to do work, that is exocytosis, releasing those synaptic vesicles from the presynaptic membrane. And then the neurotransmitter is traveling across the synaptic cleft, that physical open space, by diffusion, and getting picked up at the postsynaptic membrane where there are receptors, there are proteins that pick up that neurotransmitter. So whether it's neuron to neuron, neuron to muscle, or neuron to gland, it's going to follow that same basic structure that we looked at the neuromuscular junction. With that same sequence of events, we have an impulse that's arriving, traveling down the presynaptic membrane. It's triggering the release of a neurotransmitter from these synaptic vesicles. The neurotransmitter diffusing across the synapse, across the synaptic cleft. The neurotransmitter binds to receptors in that postsynaptic membrane and causes some change in that postsynaptic cell. So we're going to talk about what is this nerve impulse that's traveling down the axon and causing changes in other cells. This is a physiology topic, so we don't really need to know much about it. So the nerve impulse is this action potential of a nerve. An action potential is this all or nothing, self-propagating depolarization of the membrane followed by repolarization of the membrane. So it's an electrical event. It's the cell essentially acting like a battery with positive and negative charge. It's an excitation event, like we talked about in muscle fibers, due to a change in electrical charge across the membrane. So living cells have a negative charge inside and a positive charge outside of the membrane, And cells that are excitable, that have this ability to conduct the impulse, like muscle cells, like neurons, they can temporarily reverse that charge gradient. So they go from negative on the inside and positive on the outside to positive on the inside and more negative on the outside. And this change in charge is due to exchange of ions across the membrane. Ions are particles that carry a positive or a negative charge. So these cells can temporarily change their electrical charge, and that's excitability. A stimulus is anything that causes this action potential, that all or nothing electrical event, to occur. And the stimulus has to overcome the threshold level of that particular neuron. Basically, the stimulus has to be strong enough to cause that action potential, that all or nothing event to occur. So the threshold level is the amount or the level of stimulus that's required to create the action potential. And once that impulse does begin, then it's propagated, it travels all the way down the length of the axon. So it's almost like a wave of electricity traveling along the membrane, down the dendrite to the body or the soma, and then down the axon to the telodendria, to those axon terminals. Now, you don't need to know the physiology of the action potential, but do know that it's this electrical excitability event, that it's happening at the cell membrane, and that it's this one-way communication line. So I'm going to show a video from one of my favorite channels. It's 2-Minute Neuroscience, and they're going to explain about the action potential very quickly. If you want to watch it again on your own, because they do talk so fast, I would encourage you to check it out.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the action potential. The action potential is a momentary reversal of membrane potential that is the basis for electrical signaling within neurons. If you're unfamiliar with membrane potential, you may want to watch my video on membrane potential before watching this video. The resting membrane potential of a neuron is around -70 millivolts. When neurotransmitters bind to receptors on the dendrites of a neuron, they can have an effect on the neuron known as depolarization. This means that they make the membrane potential less polarized or cause it to move closer to zero. This chart shows membrane potential on the y-axis and time on the x-axis. When neurotransmitters interacting with receptors causes repeated depolarization of the neuron, eventually the neuron reaches what is known as its threshold membrane potential. In a neuron with a membrane potential of -70 millivolts, this is generally around -55 millivolts. When threshold is reached, a large number of sodium channels open, allowing positively charged sodium ions into the cell. This causes massive depolarization of the neuron as the membrane potential reaches zero and then becomes positive. This is known as the rising phase of the action potential. The influx of positive ions creates the electrical signal known as the action potential, which then travels down the neuron. Eventually, the action potential reaches its peak. Sodium channels close and potassium channels open, which allows potassium to flow out of the cell. This loss of positive potassium ions promotes repolarization, which is known as the falling phase of the action potential. The neuron returns to resting membrane potential, but actually overshoots it, and the cell becomes hyperpolarized. During this phase, known as the refractory period, it is very difficult to cause the neuron to fire again. Eventually, the potassium channels close and the membrane returns to resting membrane potential, ready to be activated again. The signal generated by the action potential travels down the neuron and can cause release of neurotransmitter at the axon terminals to pass the signals to the next neuron. Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment,
Sorry about that, folks. So we're not quite done talking about neurons. They can be divided up a few different ways. And the first way is based on their structure or their anatomy. So thinking about anatomical or structural classifications of neurons. The first one is called a bipolar neuron, because it has two poles, 1 and 1, with the cell body located between one long dendrite and one long axon. A second type is called pseudo-unipolar or unipolar, with the cell body off to one side. And what that means is that the extensions from the cell essentially function as one really long axon. So there's still messages that are received up here at the dendrites, but then the dendrites merge into essentially just one really long axon that then extends into telodendria and those axon terminals. So the cell body is off to one side of the axon. And then multipolar neurons typically have a single axon and multiple dendrites branching off of the cell body. This multipolar type is the most common type in the central nervous system. This is because neurons within the central nervous system could be getting signals from hundreds or thousands of other neurons. So they have a lot of these receptor structures, these dendrites that are responsible for receiving messages from other cells. Bipolar neurons, these are fairly rare. They are found within sensory structures. So they're found within the retina, they're found within the olfactory epithelium. And we'll talk more about bipolar neurons when we get to the special senses. Pseudo-unipolar neurons, these are primarily sensory neurons. So they're receiving information, they're receiving a stimulus, and then sending that information along one really long axon. So those pseudo-unipolar, they're sending information from the peripheral nervous system to the central nervous system. And then multipolar, they're the most common type. They're going to be motor neurons and they'll be interneurons. So they'll be within the peripheral nervous system and the central nervous system. Neurons can also be divided up according to their function or their physiology, so what they do. Afferent, or sensory neurons, are sending information from the peripheral nervous system to the central nervous system. So they're transmitting information from a sensor to the central nervous system. Efferent, or the motor division, this is sending information from the central nervous system to the periphery. It's transmitting information from the central nervous system to an effector. The last type is an interneuron or an association neuron. This is transmitting information within the central nervous system. These are located entirely within the central nervous system. They're providing that link between motor and sensory neurons. They can analyze sensory input and coordinate those motor outputs. So I'm going back to our simple model of how the nervous system works. And I told you that this top row is that sensory line, and the bottom row is that motor line. And there are three neurons labeled here: the sensory neuron, the relay neuron, and the motor neuron. And I'm going to take a minute and let you see if you can identify those three neurons based on their structural classification, what they're likely to be, and their functional classification. So take a minute to see if you can identify those three neurons based on structural and functional classification. Okay, I can't wait anymore. The sensory neuron, hopefully you said that their structure is that they're probably pseudo-unipolar, those neurons that have their cell bodies off to one side, though some sensory neurons are bipolar. So sensory neurons could be pseudo-unipolar or bipolar. if you identified it based on its functional classification, you should say that it's afferent. The relay neuron is probably a multipolar neuron, those that have one axon and a lot of dendrites. And based on its functional classification, it'd be considered an interneuron because it's totally within the central nervous system. And for the motor neuron, hopefully you said that it's a multipolar neuron. Most motor neurons are multipolar, and its functional classification would be that it's efferent. It's carrying information away from the central nervous system to an effector. And now we're adding a bit more complexity to this situation, but it's essentially the same as that model that you were just looking at. The one complication that we're adding is whether the information that's coming in is about internal conditions, so conditions from, the viscera, from the digestive system, from the heart, about the blood, or is it something that's within your conscious control that you're aware of, and that's somatic, and that's shown in blue there. So visceral pathways shown in red, somatic pathways shown in blue. So the first step for all of them would be that there's some receptor that's receiving information. It's receiving a stimulus, a change in that external or internal environment. And here I'm just showing some free nerve endings in the skin. One thing to be aware of is that receptors are essentially dendrites, that receiving part of the cell. So there's dendrites that are receiving information. And these receptors are passing information to sensory neurons in the peripheral nervous system. This is along that afferent pathway. And that afferent pathway is traveling into the central nervous system for processing. And let's follow first just those things that you're aware of in blue. That's somatic. If it's something that happens very quickly, this is something that's going to go just to the spinal cord and back. This would be a reflex. So it goes immediately from sensory neurons to motor neurons and goes along that efferent or motor pathway, causing a change in an effector. So like when your doctor hits your knee with that hammer, this would be one of those pathways, a receptor carrying information along that sensory or afferent line, and that's immediately processed and causes a change in somatic motor neurons, travels along that efferent motor pathway, and causes a change in the effectors. So that'd be a reflex. Another pathway of things that you're aware of might be processed by interneurons, those relay neurons in the central nervous system, but ultimately have the same effect, causing changes in those motor neurons along the efferent pathway. Let's say the stimulus is something like really high blood carbon dioxide. That's going to be detected by sensory neurons. They'll carry that information to the central nervous system. And in the central nervous system, it can either be processed by these interneurons, or doesn't have to be processed by interneurons, can go directly to those visceral motor neurons. And either way, those motor neurons cause a change in visceral motor neurons in the peripheral nervous system, and that causes a change in some of these visceral effectors, things that are beyond your conscious control, things like smooth muscles in the digestive system might be affecting glands, could cause a change in your heart rate by affecting cardiac muscle, or could cause the burning of fat in adipose tissue. But either way, we're getting that same basic model of the sensory or afferent line detecting some changes, then processing and integration in the central nervous system, and then this efferent motor line causing some change in effectors. So producing that action, producing the effect, often opposing that original stimulus, bringing you back into relatively constant internal conditions. So we talked about neurons I want to talk about neuroglia today, and that's the last topic that I'm going to cover. Remember, we said that neuroglia are those supporting cells of the nervous system. They're supporting neurons, and there are various types that you need to know about. The one on the left here that's blank, you don't need to know about. That's why I've removed that information. So neuroglia are found in the peripheral nervous system, so outside of the brain and spinal cord. And there are many more types that are found within the central nervous system, within the brain and the spinal cord. So the one type that's found in the peripheral nervous system is the Schwann cell. Schwann cells are what forms an electrical insulating layer around axons in the peripheral nervous system. This process is called myelination. It's this lipid-like substance called myelin, and it's sort of like the rubber around an electrical wire. It lets information be transmitted more quickly. So Schwann cells form that myelin layer around axons of peripheral neurons, and they can also help to participate in a repair process after injury to a neuron. Within the central nervous system, cells that are doing that same job are oligodendrocytes. They myelinate axons within the central nervous system. It's a totally different type of cell, but is performing the same function. They can provide some structural framework because they can link neurons together. Another cell type is the astrocytes. Astro means star, so these have a lot of projections sticking out in different directions. They're sort of star-looking cells. These star-looking cells help to maintain the blood-brain barrier, meaning they keep things that are in the blood from reaching neurons. So they control the chemical content of the environment around neurons. They help to provide structural support. They regulate the ion, nutrient, and dissolved gas concentrations. They absorb and recycle neurotransmitters, and they help to form scar tissue after injury. Microglia are these phagocytic cells. They crawl around like Langerhans cells do in the skin, and they protect the neuron by removing cell debris, wastes, and pathogens by eating them. That's phagocytosis. The last type is the ependymal cells. Ependymal cells line open cavities around the brain and the spinal cord. So they're epithelial-like tissues. They're forming this lining of a hollow space where you would typically see epithelial cells. So they're lining the ventricles of the brain and the central canal of the spinal cord. They assist in producing, circulating, and monitoring the cerebrospinal fluid. So they monitor the composition of the cerebrospinal fluid, and some ependymal cells are responsible for secreting cerebrospinal fluid. Cerebrospinal fluid is said to be a distillate of blood, meaning it's some components, but not all components of blood. So you can think about it as being really similar to plasma. It's like blood, but without the blood cells and without the blood proteins. So it's some of the components of blood, but not all. And it's a way to protect the brain and protect the spinal cord against anything dangerous that might be traveling in the blood, but still let the brain and spinal cord get oxygen, get nutrients, and get rid of wastes. So cerebrospinal fluid is sort of like the blood of the brain and the spinal cord. And here's an illustration showing these neuroglial cells, and this cross section at the top tells us that we're looking at the spinal cord. This is a transverse section through the spinal cord. And that means that we're in the central nervous system. So we're only going to see cells of the central nervous system. Let me give you a brief aside and let you know that nervous tissue is divided up into gray matter and into white matter. And we'll talk more about this next week. but I want to introduce it now. So gray matter is the cell bodies and dendrites of neurons. And because it contains the big, dense cell bodies that have the nucleolus, that have the Nissl bodies, this is going to appear gray because it appears darker because of all of those cell bodies and organelles. So with the cell bodies and organelles and dendrites, this is the site of integration. This is where messages are being received at the dendrites and processed at the cell bodies. Then white matter, shown down here, is just the axons of neurons. So no cell bodies, none of those darker organelles and inclusions in the cell bodies. It's just axons, often wrapped in myelin, so it appears white because it's just these myelin-wrapped axons. And white matter is just transmission of information. So gray matter, dendrites, cell bodies, and receiving information. White matter is just axons and transmission of information. We'll go over the distribution of gray and white matter more next week. So in this illustration, the neurons are shown colored in blue, and then the neuroglial cells are shown in yellow and in pink up here. So you can see illustrated some of these functions of the cells. So the first one is the oligodendrocyte. Remember that those are responsible for making the myelin insulation in the central nervous system. So here's an oligodendrocyte making this myelin wrapping around the axon of a neuron. And one thing that an oligodendrocyte can do is that it can insulate multiple neurons. So this one has its nucleus up here, and then it's got this process that's wrapping around this axon. And it also has this process wrapping around this axon. So it's grouping those neurons together by function. They are only found in the white matter because they only wrap axons, and axons are white matter. Other cells are the astrocytes, those star-shaped cells. So they have star-shaped branches that are out to multiple neurons, so they're touching this neuron, and they're also touching blood vessels. So here's a capillary, a very small blood vessel, and they're connecting this neuron to this nutrient supply. And you can think about astrocytes sort of as the gatekeepers, because they determine what can enter into a neuron. So they would let sugar cross across into a neuron, but they protect neurons against toxins like alcohol. So the astrocytes are the gatekeepers for neurons. And they'll be found in both gray and white matter. So here's one down here in the white matter, and here's one up here in the gray matter. Microglial cells would be found in both white and gray matter. Remember, they're roving immune cells. They play roles in repair and defense. These up here are the ependymal cells. Ependymal cells forming the lining of hollow cavities around the brain and the spinal cord. They're epithelial-like cells, and together with capillaries, they're responsible for producing the cerebrospinal fluid, that distillate of blood that provides nutrients, provides oxygen to neurons. So the cerebrospinal fluid would be traveling in the central canal of the spinal cord, and that's the central canal right there. So it's this distillate of blood. We're not seeing Schwann cells here because Schwann cells are only found in the peripheral nervous system, and we're looking at the central nervous system. I want to talk a little bit more about myelination, and there's myelin sheath. So you know that nerve cells, neurons, shown here, have this really long extension, a long extension that's called an axon that's responsible for transmitting information. myelin, this insulation layer, forms around the axon in what's typically called the myelin sheath. So these axons are sort of wires sending electrical signals to different parts of the body. And these axons are connecting neurons to other cells, including neurons, muscle cells, glands at those sites called synapses. And the myelin is composed of these support cells that protect the axons. So it's sort of like the insulation on electrical wires. Myelin lets nerve cells transmit information faster and allows for more complex brain processing. And in the central nervous system, those oligodendrocytes are what's responsible for forming this myelin sheath, and they can wrap around multiple axons. In the peripheral nervous system, Schwann cells are what wrap around the axon, and they can't connect multiple cells because their entire cell body is wrapped around one axon. And what happens is that you have a Schwann cell that starts wrapping around an axon, and then you keep wrapping and wrapping and wrapping. So these are layers and layers of cell membrane of the Schwann cell out here. And eventually, you end up with just the nucleus squeezed off to the side and layers and layers and layers of insulating cell membrane. And that's what makes the myelin sheath. So the myelin sheath occurs in the central nervous system with oligodendrocytes and in the peripheral nervous system with Schwann cells. Myelination starts happening in utero when a fetus is about 16 weeks old, and it continues all the way into adulthood. So myelin sheaths are developing as someone is growing and developing. One of the reasons that small children seem so clumsy is that a lot of their axons are not yet myelinated, and that makes them have slower response times and less coordination. So when small children seem really clumsy, it's because their nervous system hasn't fully developed yet. If you notice, that you're interacting with tweens and early teenagers, this is where myelination is particularly occurring in that frontal lobe of the brain, and it's helping with cognitive development. So myelination continues throughout development up until early adulthood. Not all axons have a myelinated coating. So even though the myelin lets impulses travel faster, that's not always a good thing. Firstly, there's not enough space inside the body to have every neuron axon wrapped up in insulation like this. If you had every axon wrapped in these myelin sheets, nerves would take up a lot more space. You wouldn't have as much room for everything else in the body. So one thing is just a space factor. Second, you need some nerve impulses to travel more slowly. So let's say that you hit your finger with a hammer accidentally. What you feel is a sharp pain right away. That's those myelinated axons that are helping you respond more quickly. So the sharp pain, that's going to be myelinated axons. But then for a while, you'll feel sort of a dull thudding pain that continues. That's those unmyelinated neurons carrying that information more slowly. And that's helping you learn and telling you to protect that damaged hand. So there's a role for both myelinated and unmyelinated axons in the nervous system. So we've talked about neurons, and we've talked about glial cells. And I'm going to give you just a minute or two to see if you can match each neuroglial cell with a description of its function. And I'm going to start giving away answers. If you're not quite done, you can pause the video. So oligodendrocytes, remember those are the ones that are forming the myelin sheath in the central nervous system, and they can wrap around multiple neurons and connect them according to function. Astrocytes, those star-shaped cells, they're the ones that anchor neurons and blood vessels. So they're connecting neurons and blood vessels, acting as a gatekeeper. They're maintaining that extracellular environment around neurons, keeping them safe, assisting in the formation of the blood-brain barrier. So protecting the brain and the spinal cord from anything dangerous that might be traveling in the blood. Microglial cells, those roving phagocytic cells in the central nervous system that eat pathogens, eat debris. Schwann cells help to create the myelin sheath in the peripheral nervous system. And that just leaves ependymal cells, those ciliated epithelial-like cells in the central nervous system that are responsible for forming and circulating the cerebrospinal fluid. So if you have any questions, comments, concerns about this, please let me know. Quiz 6 is going to be on Monday, and it's going to cover the endocrine system and what we talk about today from the nervous system. So please let me know if any questions arise.